Good morning, students. The topic number 4 in the genetic engineering is the history and milestone in the rDNA technology. Today, I will focus on certain historical achievements and landmarks in the field of genetics and how the pathway of rDNA technology has designed by the various discoveries and inventions made by the scientists by the time. So we already discussed numbers of scientists and their discoveries in earlier lectures. Mendel, Sutton-Boveri, Muller, Mesher, Morgan, Griffith, McIntosh, Biddle, Tatum, Avery, McLeod and McCarthy's work, Hersey and Chase, Watson, Crick, Jacob and Monod. These all are the fellows or the scientists who discovered and invented many things in the understanding of genetics. Initially, we see some such important events in this genetics, including the work of Gregor John Mendel, who we know as the father of classical genetics, who gave the first concept of the chromosome, the Mesher, who isolated a nuclei, separate the nucleic acids from the various cells and called him is a nuclein. Richard Altman, who purified protein-free DNA in 1889. In 1909, the word biotechnology is used in print for the first time. So these are the history. In 1923, the Griffith, who shows the bacterial transformation in which the DNA is transformed from one cell to another cell. So he proved this through experiment on a mouse. Then in 1934, it is demonstrated the bacteriophages are composed of DNA and protein. In 43, Luria and Dalbrook experiment shows the mutations occur in microorganisms including viruses and bacteria, and bacteria lost the attachment of bacteriophage due to mutation. Oswald Avery, McCarthy and McLeod, these three persons showed that the genetic material is DNA, not the RNA or protein or carbohydrate or lipid. Salvador Luria, which shows that DNA repair themselves naturally. Rosalind Franklin, the scientist who gave the major contribution in understanding of double helix structure of DNA through X-ray differentiation data, and shows that the DNA is a double helix as well as in a spiral form. Edwin Chargaff, who gave you the concept of complementation. The base pairs are complementary to each other and they are in a proper ratio. A bind to T and G bind to C. That thing has been shown by the Chargaff. Hersey and Chase, this shows that DNA is the genetic material in bacteriophage also, not only in bacteria but in virus DNA is also a genetic material through his famous blender experiment, where he used P32 and S35 radio label isotopes. In 1956, it is demonstrated that RNA can be a genetic material, and first time observed in TMV, tobacco mosaic virus. RNA can be a genetic material. Arthur Korenberg. So whatever the discoveries which are common in the genetics has been discussed. Now we are discussing the milestone in rDNA technology. This milestone means those researches which make this rDNA technology possible. And today there is a great advantage of such discoveries and inventions. So first in that category in 1956-7, Arthur Korenberg, they synthesized DNA test tube after the discovery of DNA polymerase 1. So DNA polymerase 1 is the enzyme for the synthesis of DNA. So this is the most important enzyme for DNA replication, and we need these enzymes in multiple tasks. In 1957 or 8, R.W. Hole, Nirenberg and Hergovin Khurana, they find out the tRNA molecule with Francis Crick. They propose the tRNA structure and how it works. Lederberg, who got the Nobel Prize for the discovery of bacterial recombination. So just I am giving you highlights of this one. The lecture on this scientist is already given to you. This thing was not in our previous lecture. This discovery of penicillin. So discovery of penicillin is not related to any genetics because it is in the field of medical. But the discovery of penicillin nowadays it is useful in genetic engineering for the development of screening technology. To find out the mutant or recombinant, we are using the media containing antibiotics. So this is the gradient plate technique which was discovered by the Skybalaski, where antibiotic is used. Jacob and Monod, they discovered the operon. Francis Crick proposed the Hubble hypothesis for anticodon. Jacob and Monod, they discovered the genetic control of enzyme and virus synthesis in 1961. These are most important discoveries which make the technology now in an advanced stage. Nirenberg, Leder and Khurana cracked the genetic code using RNA homopolymer and heteropolymer experiments in 1966. In 1967, the DNA plasmid were isolated and purified by Vinogradov. R.W. Hole, Khurana, Nirenberg, the same thing is repeating over here. Discover the genetic codes. Hersey, Dalbrook and Luria find out the replication and recombination in bacteriophage. In 1969, for the first time, the enzyme was synthesized in test tube outside the cell, we can say cell free extract. So whatever the things occur within the cell, the same things performed in a test tube. It is known as the cell free extract and cell free synthesis reaction. So this is a new for you. In 1969, the enzyme was synthesized for the first time. Hamilton, Smith and Delian Nathans and Werner Arbor. You have to remember these three scientists. For what purpose? Hamilton Smith, Daniel Nathans and Werner Arbor, for the first time they discovered the restriction endonuclease, and from this discovery the recombinant DNA technology concept was start to think about, is it possible to cut the DNA at desired site, is it? So this is the most important discovery in the field of rDNA technology, discovery of restriction endonuclease by these three scientists, Arbor, Nathans and Smith. So you can see in the figure the restriction enzyme cut the DNA at specific site, and that site is a known for that enzyme. So there are 600 700 different kinds of restriction enzymes for yeast, bacteria and fungi and algae. Temin and Baltimore. This is second most important discovery in the field of genetic for rDNA technology. That is, these two scientists discovered the activity by RNA virus, that is reverse transcription. Reverse transcription is the phenomena where the DNA is synthesized from RNA. So this is performed naturally by RNA virus. When this RNA virus enter in a bacteria, then the RNA first convert into the DNA, and this is found in the Rhesus sarcoma virus, and this is possible by the action of reverse transcriptase. So it is a very important discovery. You have to remember for exam. Temin and Baltimore discovered the reverse transcriptase enzyme. So reverse transcriptase enzyme is reverse reaction of DNA to RNA. The reaction is RNA to DNA. So the DNA that is produced by the activity of reverse transcriptase, it is known as cDNA, complementary DNA. Then Stanley Cohen and Herbert Boyer. These two scientists are the father of recombinant DNA, or father of cloning, we can say. Stanley Cohen and Herbert Boyer and at Stanford University first constructed the recombinant DNA, and this recombination is possible with the work of restriction endonuclease. They cleave the DNA by this enzyme and attach the two different pieces of DNA and join them with the DNA ligase enzyme. So they are known as the father of recombinant DNA technology, Norman Cohen and Herbert Boyer. That time in 1973, the first time the company named Genentech Biotechnology, the world's first genetic engineering company, was founded. Now we are discussing the important research technologies, important technology for rDNA 1. So it including the Southern blotting technique. E.M. Southern developed this method for recovery of DNA fragments from electrophoretic gel. So we have a last discussion in unit number 3 on blotting techniques. In 1975, George Kohler and Cesar Milstein developed the technique known as the monoclonal antibody. What it is monoclonal antibody? In monoclonal antibody, an antigen is given to the mouse and that mouse produce antibody. So these are the B cell. The B cell are responsible, they are the type of lymphocyte, B lymphocytes, and these B lymphocytes produce the antibody in vertebrates. So these are the antibody producing cells and they are fused with cancerous cell. Cancer means a cell which grow uncontrollably. Their control is lost. So this excessive growing cell are fused with antibody producing cell. These cells are known as the hybridomas. These cells are known as the hybridoma. Hybridoma cell is the fusion between the myeloma cell and B cells. Myeloma means cancerous cells which divide uncontrollably. They divide uncontrollably. Their cell division is continue without any restriction. And then they are fused with the B lymphocytes. So B lymphocytes are responsible for antibody. And this way, the huge amount of antibody are produced. So these antibodies are produced are known as monoclonal antibody. This technology is used for production of single type of antibody production in a bulk. So this is known as the monoclonal antibody. It is important for vaccine development and antibody production. The Pribno box, which is found out by starting of the transcription. Okay. Each gene sequence is expressed in E. coli for the first time. In 1976, the barrier of eukaryotes and prokaryotes has been removed and yeast cell expressed in a E. coli. In 1976, this is most important discovery, that is DNA sequencing, and right now has a great value. So this DNA Sanger method for sequencing is today a widely accepted method and given first time by this Sanger and Charles Colson. So this technology give you the idea about DNA fragments. Using this technology, we can find out the sequence of DNA. In 1978, in vitro fertilization, IVF, Louis Joy Brown was born the first human baby resulting from the in vitro fertilization. In vitro fertilization means the female egg is recovered from female urethra, from female, and it is transferred in the test tube. And in the test tube, the sperm cells are added and allow them to fertilize. After fertilization and few cell division, the fertilized egg again implanted in the mother womb. So that mother hatch the baby normally. So this in vitro fertilization technology was invented in mouse and now it is for the baby development normally for human. So this is very common nowadays in vitro fertilization in humans. Dr. Hargobind Khurana, who reported the complete synthesis of a biological functional gene in a test tube. So we can see over here such advanced discoveries since 1975. You will see that all the technologies on a advanced stage. In 1979, Alvin, Kemp and Stark at Stanford University developed northern blotting technology. Northern blotting technology is the name given from the southern blotting technology. Southern blotting technology is for the recovery of DNA and this one is for the mRNA. This mRNA attach on the paper, that paper is chemically reactive paper, separate paper. So this technology developed by these three scientists. Then after Taubin developed the Western blotting technology. This Western blotting technology is for recovery of protein. So for protein, for DNA, for RNA, for other substances, there are various blotting techniques has been established through the time. So you can see the basic one is the southern. The first method was the southern technique for DNA. Then after for RNA and Western for protein. Then after, to understand the protein technology interaction, far western, protein DNA, then southern western, lipid can be possible to identify far eastern, middle eastern for DNA and RNA, post translation modification, eastern for RNA and protein interaction, northwestern. So this way the various blotting techniques were discovered. Paul Berg, Gilbert and Sanger developed the method of gene mapping, DNA mapping through the recombination process. So this was the important discovery at that time, gene mapping. And they construct the recombinant DNA molecule containing the Fas lambda gene and inserted into small circular DNA molecule, that is the plasmid. So this scientist developed the cosmid and viral gene inserted into the bacterial gene, plasmid. So true rDNA technology was initiated in 1980. Same time, Ohio University students made first transgenic animal. You can see the cow, which is a transgenic animal made by Ohio University in 1980. In 1981, the first transgenic animal produced by transforming the gene from other animal into the mice. You can see in the photograph, that is a transgenic animal. The first patent for genetically modified organism is granted for the bacteria that can break down crude oil. This achievement was made by Dr. Anand Chakrabarti, who is the Indian origin scientist, and got the patent on Pseudomonas putida, which he constructed by combinations of various genes into the plasmid, which cleaves the oil, and that for the removal of the oil in the sea. So through activity, there are huge amount of oil spillage occur into the sea. દરિયામાં બહુ બધું ઓઇલ ઢોળાઈ ગયું હોય હજારો કિલોમીટર સુધી, and that is degraded by this bacteria Pseudomonas putida, and Dr. Anand Chakravarti first get the patent on this one. In 1980, Stanley Norman and Cohen Boyer, who developed the genetic engineering technology, and they got the first US patent for gene cloning technology. And by providing the successful outcome of cloning, they express the insulin DNA into the E. coli. And E. coli produce the insulin for the first time, which was produced by Eli Lilly company. The foreign to unicellular organism, that is a foreign protein. So that foreign protein discovered by these two scientists introduced into the E. coli, and afterwards they used the similar technology for human growth hormone erythropoietin. This patent earns $300 million for the Stanford University. In 1982, after 2 years of discovery, US FDA, United States Food and Drug Administration, released this insulin for the human purpose. So the question is asked, what is humulin? So humulin is the genetically produced insulin produced by Eli Lilly company. In 1983, Barbara McClintock, who find out the transposable element or transposon, or we can say jumping gene. So they are used for the transfection in today's science. Cell Chilton von Montague and Horst, they find out the working of Agrobacterium tumefaciens gene transfer. So there is a great importance in plant biotechnology of TI plasmid. So these scientists find out how these Agrobacterium tumefaciens use the TI plasmid and infect the plant. So we will see in detail. Kerry Mullis give the most important technology, well-known technology nowadays. There is a PCR, polymerase chain reaction. So this polymerase chain reaction techniques which amplify your small piece of DNA in hundreds to thousands to lakhs of the copies in few hours. In this technology, the temperature is maintained above 60 degree throughout the process, 60, 70, 90. So normal polymerase cannot work. For this purpose, the special polymerase is required. You have to remember this polymerase enzyme. Thermophilic organism is there. Their polymerase name is Taq polymerase. Taq polymerase. Taq means Thermus aquaticus. Thermus aquaticus, the name of organism from which the enzyme DNA polymerase is taken, which can withstand at the high temperature. It is a thermostable enzyme. So PCR, polymerase chain reaction. We will see this PCR in detail in coming lectures. In 1985, DNA fingerprinting. DNA fingerprinting. It is nothing but the RFLP. Restriction, fragment, length, polymorphism. In this technique, the whole DNA of any organism is broken down by a single kind of restriction endonuclease enzyme. So whole DNA is broken down into the small piece, and then after This DNA is run in electrophoresis, which creates a band. So this band's impression is known as the DNA fingerprint. On the screen, what are you watching? The barcode pattern. That barcode like pattern is nothing but the DNA fingerprint. Then in 1985, the instrument developed, that is automatic fluorescent sequencer. This fluorescent sequencer instrument is the instrument which gives you the sequence automatically. So this was the advanced instrument in this era in 1985, gene sequencer. Same time there was a development of the gene gun. Gene gun used for particle bombarding. The DNA attach on some gold particles, and these gold particles are shooted from this gun onto the target cells. So DNA directly enter within the DNA nucleus, plant nucleus. So this way, this is the mechanical method for gene transfer. This is the physical method of gene transfer, and the technology is known as the particle bombardment. In 1986, the first recombinant vaccine is approved for human use. After insulin, the most important discoveries, successful technology was hepatitis B surface antigen. Normally, hepatitis is the virus, and the virus is used for the vaccine production, which may cause the disease if it is improperly treated. So, to remove this threat, instead of taking the virus, only its protein DNA is used for the development of vaccine. The first anti-cancer drug, interferon, was also developed in that time. So such products were started to produce by technologies in 1985-1986. The Dolly cloning of the sheep was possible in 1996. The genes from two different parents taken, that is fused. So the development was the Dolly. When the scientists are Wilmut and their colleagues, they did this successful experiment at Roslin Institute in the Scotland. In 1987, the first field test of genetically modified food plant was approved for virus resistant tomato. And in 1994, genetically modified tomatoes are sold for the first time in the United States, US. In 1996, the sequence of Baker's genome is completed. So then after the time was come where the whole gene sequencing was started for organism. First viruses, then bacteria, and now yeast was gene sequence in 1996. In 1997, the techniques was developed DNA microarray. The microarray is the miniaturization of the techniques where the various principles of antigen-antibody binding, fluorescence, radiotope isolabeling used in miniaturization system where the genes are identified for any organism. So here the silicon chip or microscopic slide is used, and this is the very space saving and highly specific technology was developed, microarray. Then in 1997, East artificial chromosome was designed by the Olson at Washington University, which is used widely for human genome project. This artificial chromosome carry very important gene and used for the construction of gene library. In 1998, the human erythropoietic stem cells cell line has been established, which can use for the disease or dysfunctional cells. So we know that the stem cell, the cell which is the very foundation cell from which any part of the body can be regenerated. We know that at the time of our birth, only single cell zygote is developed from oocyst and sperm fusion. So that single cell is known as the erythropoietic stem cell. This erythropoietic stem cell has the capacity to develop the whole body because it is the totipotent stem cell. Totipotent means totally potential. So this totipotent stem cell have the information for the development of any part of the body, and this is useful for the therapy at any age. If a person have a cancer of any cell, so we can find out the gene or we can utilize this stem cell. Nowadays there is a stem cell repository bank established. The placenta, immediate after the birth, the placenta જેને તમે ગર્ભનાળ કહો છો બાળકની, that placenta is preserved into the placenta bank, and this placenta preserved bank will give you the your preserved baby's placenta when it is required. They maintain for a life of a person. So this kind of preservation bank was initiated in 1998. In 1999, the next gen gene sequencer has been established. We know that the Human Genome Project has took at least 13 years to complete. But if the same project was run by this instrument, it can complete the Human Genome Project within 6 months, not 13 years. So this much speed was nowadays available with more advanced gene sequencing machine. In 1999, the capacity of this sequencer was 76,800 base pair per hour, which is the 5 to 30 times faster than that existing sequencing. So if Human Genome Project run on this instrument right now, so it will take only 6 months to complete. In 2003, the technology is initiated for the identifying the viruses. SARS, severe acute respiratory syndrome, a virus. So when this virus arose in the China, its gene sequencing was made within 3 weeks. So this is the advantage of rDNA technology. And right now, whatever the test of corona are made, that is on PCR, and real-time PCR is used. So one time the technology was a dream. Right now it is a very common technology. In 2000, the full genome sequence of Drosophila melanogaster was completed. In 2003, Human Genome Project, which was initiated in 1998, Okay. So now in the 2003, that precise version of human genome was developed, 99.999% accurate, is published. So this is the chronology of RDNA technology, this human genome project. We will discuss the human genome project in one lecture separately. So these 13 years of human genome project have a many important landmarks. In the photograph you are watching the whole set of chromosomes. For one chromosome there are books of language, and language you can see only 4 letter A, T, G, C. The first clone pet, a kitten, is delivered to its owner. She is called copycat. This is the copycat genetically modified organism. The same organism developed by the scientist which you see. The gene is taken and copy the gene and amplified, and cloning was performed, and same cat was developed in 2004, copycat. Then this technology was useful in development of many other drugs and antibodies, vaccine, antigen development. A vaccine for human papilloma virus, HPV, for treating the cancer of cervicals. Okay, so these cervix, or not cervical, cervix. The vaccine is made by rDNA technology, and virus gene was introduced in the DNA of Baker yeast. In 2007, from embryonic stem cell, the human skin was developed. Scientists developed the human skin from the stem cell in 2007. In 2008, Japanese scientists created the first DNA molecule which made entirely artificial part. So synthetic DNA used for artificial part development. Then after the scientists construct the synthetic functional genome, whole DNA of an organism, of a bacteria, was designed in a test tube as they require. So this way the era was started. Now we can tell that era is a synthetic biology. In 2010, the researcher first developed the first synthetic cell. They developed the synthetic cell. Then after, the synthetic or artificial blood was re-engineered and produced. Then after, 3D printing technology was developed. In 3D printing technology, instead of ink, the cells are used, and the cells shape the organ. First time in 2011, the human trachea from stem cells was prepared for 3D technology. So this is the current era of genetic engineering, and we can synthesize the organs, particular size and shape, in a 3D form. In 2013, the US Supreme Court ruled that naturally occurring gene cannot be patented. And this era will come where every student bring their own transgenic animal into the classroom. So this is the completion of the history and milestone or historical achievements of rDNA technology. Thank you very much.